Namaste students. Today I am going to discuss about the topic microbial life cycle, microbial growth and their life cycle. So what is the meaning of microbial growth? In terms of microbial growth, microbial growth means the microbes increase in their size, increase in their number, means they multiply. Okay, uh, the growth of most microorganisms is occurred by the process of binary fission, in which they divide into some equal cells. In the first step, they go to DNA replication, cell elongation, then a septum is formed between the cell. Okay, after formation of the septum, this cell will divide into two equal daughter cells, each containing genetic material. So that all the cell particles, including the genetic material, is divided into two some equal parts. That is the known as the cell division or cell separation. So microbial population have some growth phases, which we say is the life cycle of the microorganism or the life cycle of the bacteria. Okay, so how we can calculate this life cycle? So this life cycle is actually calculated in terms of the time, and actually we calculate this life cycle when we prepare a culture media and we inoculate uh, the microorganism into that culture media. And when the growth originate, we can calculate the life cycle. We can analyze the growth cycle of the bacteria. So what is the growth cycle? In the bacteria or most of the microbes, there is a four types of phases. Okay, lag phase, exponential phase, stationary phase, and the death phase. Okay, so this is the lag phase. Okay, this part in this curve, this green part, in this curve, this part. Okay, this part is known as the lag phase. So what is the lag phase? Lag phase is the phase in which the cell prepare itself for elongation, for uh, division. Okay, cells are not divided in this phase, but they start to grow, they start to increase in their size so that they can multiply. That is their uh, preparation phase. So no multiplication is observed in this leg phase. Okay, after completion of this leg phase, and the microbes go to the exponential phase. Okay, in the exponential phase, what is happening? Exponential phase is the phase in which microorganisms multiply in a random phase, means they multiply. This is a very important phase for the microbial growth, and we can calculate the growth of microorganism in this phase. That is very important for the growth cycle of the microbes, because the nutrients is available in full time. All the nutrients are available because it is not utilized yet. Okay, the growth media is not utilized yet, so the microbes will multiply again and again, and they will multiply from 2 to 4, 4 to 8, 8 to 16, 16 to 32. So they will multiply. Okay, so this is the multiplication phase in which the media is very high in its content, and all the nutrients are very fresh in this phase. So cell will divide very efficiently, and they will divide very fast in this phase. It is very short period, but in this short period, we can see here the time is very low. Here we can say here, see here, that the time is very low here, but The growth is very fast. This is the this is the concentration of organism in single ml of media. Okay. For example, it was 5.2 in the lag phase, but at the starting of the uh, at the end of the exponential phase, it is approximately 8.2 per ml, 8.2 million per ml. So we can calculate it in terms of the amount of bacteria in per ml. So after completion of the exponential phase, there is a next phase that is known as the stationary phase. Stationary phase is the phase, that third phase. In the stationary phase, the media it started depleted. Okay, because it is now not a fresh media, because most of the microbes have been started multiplication and they have divided, so they need more and more nutrients. If they need more and more nutrients, then if some microbes which cannot utilize the media will start to go into the death phase. So there is a level which is parallel in the growth and in the death. So the number of bacteria which are growing is equal to the number of bacteria which will die in the stationary phase. So it is a very flat phase. It is a very flat phase in which the death and the growth are equal to each other. And after completion of this stationary phase, the death phase will arrive. In the death phase, the bacterial cell and the cell will start dying, because the nutrients have been utilized by the microbes, so they will start to go in the death phase and they will die after their particular age. So that is all about the microorganism and their growth cycle. So in the next video, we are going to discuss about the environmental effects. So our lecture is over here. Thank you very much.